So at the 2020 virtual ASCO, had the pleasure to present the data from a randomized phase 3 by the name of SAVOIR that address an unmet medical need, and that's metastatic, locally advanced papillary renal cell cancer, which is the second most common type of kidney cancer after the conventional clear cell type. And that hard-to-treat tumor is associated in some parts with alteration of the MET genes. Around 30–40% of patients have alteration of the MET pathway, whether chromosome 7 trisomy, and that's where the MET gene resides, or MET mutations, or MET amplification, or actually the ligand hepatocytic growth factor amplification. So we looked at this the population that is MET dependent, and we evaluated them in this phase 3 trial of SAVOIR versus standard of care sunitinib. This built on prior preclinical work, phase 1, as well as a phase 2 study, single arm with savolitinib, that showed 18% response rate if you are MET dependent, you have these alterations, versus 0% if not. So that led to SAVOIR. We aim to screen around 400 patients to randomize 180 patients. Unfortunately, what happened is at the same time we ran in epidemiological studies, assuming that the outcome, like progression-free survival with sunitinib in MET-dependent patients, would be quite poor, but this did not end up panning out, and MET-dependent patient in that epidemiological study did as well as the MET-independent patient. So at that time, went back to the drawing board, and we needed a much larger study to detect differences. So the decision was to stop after 60 patients. Interestingly, after 60 patients, there was a numerical difference in the outcome. Median progression-free survival with savolitinib, the pure MET inhibitor, versus sunitinib was different. The hazard ratio was 0.71. For overall survival, the hazard ratio was 0.51, all favoring savolitinib. In terms of response rate, 9% with sunitinib, 27% with savolitinib. And what's interesting is the side effect profile overall favored savolitinib over sunitinib. So we cannot draw firm conclusions at all because of these small numbers, but we are, with the sponsors, taking a seat back and thinking about next step and how to evaluate savolitinib in papillary renal cell carcinoma next. The question you ask is extremely important. To move forward, should we still move forward with single agent in a MET-enriched? Definitely in a MET-enriched population, in MET-dependent. So those alterations I talked about in 30–40% of cases. But I think, should we also think about combining them, let's say, with immune checkpoint blockers? This is a possibility we're discussing. There is a trial from the UK led by Dr. Tom Powells called Calypso. It's in all papillary RCC that combine durvalumab, the PD-L1 inhibitor, with savolitinib, and it showed some efficacy. No control arm, so it's hard to know what's contributing to what. But I think that is also a possibility in combination. Why? Because immune checkpoint inhibitor as a single agent have shown activity in papillary RCC in general.